Hello, hello, hello. Welcome, everybody. Welcome to the stream. Hello, welcome. Hello, James. Hello, Hero. Hello, Ari. Hello, someone will not see our Smith. Archie, ISO, Seed, Surprise Pants. Welcome, welcome. Welcome to you all. Hello, Penbu. Welcome, welcome. Yeah, today we are going to be talking about this year's Nobel Prize for Physiology and Medicine, which is going to be a very, very exciting talk because I am of the firm belief that, Nobel Prize winners, for as brilliant as they are not doing anything that your brain can't do, right? The difference between you and a Nobel Prize winner is that they did the thing, but I actually don't think that uh, that their brains are all that much different than yours. And what do I mean by that? Do I mean that you can win a Nobel Prize? I mean, maybe one day you can win a Nobel Prize. But what I'm saying is that when you get into the mind of these Nobel Prize winners, you get an appreciation for what brilliance actually looks like. And I don't think brilliance is this like big brain, high function mega mind thing that you'll never be able to do, but rather insight from being steeped in their research for so long. And by walking you through the background behind at least Shimon Sakaguchi's paper, I think you'll come to appreciate the nature of a Nobel level brilliance, and I think that's going to be very, very fun. But before we get into it, let me just go ahead and catch up on chat here. Physiology Nobel Prize 2025 really has me deliberating between immunology and chemistry for a second major. I think immunology has a lot of discovery potential left. I don't think we know even 30% about how the immune system works. It is so complicated and interacts with all sorts of other body systems in such unique and interesting ways. But of course, I am just a tad bit biased being an amateur immunologist myself. Suppressor T cells are like the stuff that tells you your immune system, you'll please stop killing everything. In a nutshell, that is where we are heading, but I think it is more interesting to talk about not exactly what they are, but how they were discovered. Yeah. All right, so let's go ahead and try and have a peek into this Nobel Prize here. Okay, so the physiology prize this year went to the discovery of a specific subset of T cells, the discovery of a specific subset of T cells called regulatory T cells. But before we get into specifically what regulatory T cells are, we first have to talk about what T cells are. So T cells are lymphocytes. These are cells that make up the majority of the lymph, the body's drainage system, and they originate in the bone marrow and then mature in the organ known as the thymus. Hey, Frost, welcome. T cells have incredibly diverse functions and cell types. So when I say T cell, I should really be more clear that T cells themselves contain a lot of diversity within them, right? And let me know whether or not my mic is okay. I can see myself peaking occasionally, which is not good. But yeah, I want to make sure that the audio is listenable. And if you've watched my videos, you know that T cells are a common feature in my videos, right? These are soldiers of the adaptive immune system, but not only are they soldiers, they also complete a lot of other different functions, right? So what are they? So T cells can actually mature into many different cell populations, and this is not even extensive, right? These are some popular ones to talk about. The most popular in my videos are these CD8 cytotoxic T cells. These are the killer T cells. These are T cells that are specialized for combat, right? So they get close and personal up to infected cells and can detect whether or not a cell is infected, destroy that cell, thereby denying a bacteria or a virus the ability to replicate inside of them. So these are combat-focused cells. You also have helper T cells, that are responsible for support, logistics, and shot calling, right? These are the dendritic cell gets called the general of the immune system because of its like more coordinating ability, while the helper T cell is also somewhat of a general. You can think of them as directing the immune response, helping B cells mature, helping activate macrophages and other T cells, etc., etc. Bacteria duplicate inside human cells depending on the bacteria. There are intracellular bacteria like tuberculosis that makes a home within macrophages. Not all bacteria are intracellular, but some of them are. And then we have memory T cells. So these T cells are the ones that are responsible for your long-term immunity. They kind of exist after an initial infection, and when you get infected by the same thing 2 times, they will instantly recognize that they've seen this before and then rapidly clonally expand, thereby giving you a long-term immunity to certain types of infection. Also, rabies. I thought, yeah, rabies is a virus, though. So there are different, that's why I say intracellular pathogen as opposed to specifically bacteria or viruses as a whole. Yeah. All right. So this year's Nobel Prize goes to the regulatory T cell, which is another category of T cell that doesn't get as much talk in my videos in particular. So these cells are responsible for tuning the immune response, and they play an important role in fighting infection and cancer, and they also play one more very important role. And I'm wondering if chat kind of, I don't know if you've read about this discovery before, but I'm wondering if you have a sense for why this cell is important beyond infection and cancer. And so to kind of jostle your creativity, I want to first, if you know the answer as to what these guys are primarily responsible for, don't contribute yet, but rather, why don't we take a moment to brainstorm reasons why a very weak immune response and a very strong immune response are potentially not great. Like, why does the immune system need to be tuned to begin with? So let's go ahead and think about that. Why do we need a cell that tunes the immune response? It surprises me that some infections are worse the next time you get infected, aka in dengue or chickenpox, or so I've heard from word of mouth. So chickenpox is not necessarily the worst the next time you get infected. Chickenpox hangs out in your neurons and can get reactivated as shingles. Which is pretty complicated. But I think the reason why chickenpox and shingles look different, and that why shingles is more severe, is shingles occurs when chickenpox wakes up in the neurons. So it is the same virus, but in a different location. And if you've watched my rabies video, you would know why hanging out in neurons is a little problematic. So I shouldn't say anything if I already know the answer. Yeah, for now. I just want to first lay the not ideological, but theoretical groundwork for why the T cell is important. Ramp up and ramp down based on feedback signals on the severity of disease and or disease progression. Excellent answer, right? You don't want to overreact. You don't want to have an overwhelming force deal with a relatively minor problem. And why might that be? Like you've got the right answer, but I want to hone in on exactly why this is important for the body. They're very, very lethal killing machines. Yeah, definitely getting closer, right? T cells are lethal, but of course you want them to be lethal because you want them to kill things like bacteria, like viruses, like cancer cells. So why do we need a mechanism to tune the immune response? I had shingles once inside the ear canal. I didn't even know that was possible. Yeah, that is interesting. That is interesting. Like how neutrophils overreact in Ebola. Yeah. So what does overreaction of the immune response lead to? What does overreaction lead to? To control collateral damage. Exactly. Overresponse can cause more harm than good. Exactly. You want your immune response to be measured. You do not want it to go too far, because something that is capable of killing bacteria are, they're often using mechanisms that can kill your own cells as well. So a regulatory response is pretty important. Okay, so here's the thing. Not only is it important that the immune response be tuned appropriately so that you don't get collateral damage because of mechanisms that are non-specific, but also having an immune response that is uncontrolled can lead you to develop an immunity to yourself. And that is pretty stressful. Right? As you can imagine, if your immune response is so easily triggered that even your own proteins are going to trigger it, That is a problem. Can I answer already, or is it too soon? Well, we will get into it together. As you can see, the answer that I'm fishing for here is right on screen here. We have to talk about autoimmunity. So regulatory T cells are important to tune the immune response, particularly because your immune response isn't perfect, because your immune response can be trained on things like bacteria and viruses, et cetera, et cetera. But why doesn't your immune response affect yourself? And there are several reasons, right? You know, the immune response was trained to recognize non-self, so foreign antigens like bacteria, viruses, fungi, whatever, and discriminate from them against the proteins that you make. Because if they reacted to the proteins that you yourself made, well, you would essentially be consumed from the inside out, right? And that would be devastating. You don't want that to happen. And so how is autoimmunity prevented? And I believe I went over this in one of my videos of I think it was maybe the microbiome video, but one of the ways that autoimmunity is regulated is through a process called negative selection. And so negative selection occurs in the thymus, the particular immune organ that causes, or the particular immune organ that houses maturing T cells. And so these specialized cells called MTEC cells specialize in training these T cells, showing them host antigens. And so if a T cell does not react to your protein, it gets to live and mature and live out the rest of its life as an active T cell. But if that T cell does react to the host protein, apoptosis is triggered and that T cell doesn't get to exist. So does a lack of suppression cause allergies or autoimmune disorders like psoriasis or lupus? So I'm not fully brought up to speed on the mechanisms of any of those, allergies, psoriasis, or lupus. I think lupus is autoantibodies, so I'm not entirely sure how B cells work with the autoimmune machinery, but I wouldn't be surprised, is what I would say. Okay, so that's negative selection, right? But the thing is, if negative selection worked 100% of the time, we wouldn't have this idea of autoimmunity. I don't know if you can hear the garbage truck outside. That's super annoying. We're just going to have to truck along here. Yeah, so if negative selection worked 100% of the time, we wouldn't have the concept of autoimmunity. So because biology is just weird shapes bumping into one another, this process of training T cells is often not perfect. So sometimes immune cells can be trained incorrectly. Sometimes during the training regimen in the thymus, some T cells that react to host cells are going to be allowed to escape, which is kind of annoying, right? Like this filter that we learn in school, if you've taken a basic immunology class, this filter that's supposed to exist to make sure that you don't produce an autoimmune reaction isn't perfect. And so you evolved these cells called T regulatory cells that ensure that when immune training goes wrong, that the immune system does not destroy the body, right? All right. Yeah. From my explanation, it does sound like allergy existing would be a failure of negative selection working perfectly. So I know that in the allergic response, there's a separate cell that mediates the allergic response. I can't quite remember, maybe basophil. I actually don't remember what the immune cell that eosinophils are parasites. Mast cells are the ones that deal with the immune response to allergies, at least one of the cells that do so. So I'm not entirely sure how Tregs play into allergies. I don't want to make a claim that I can't actually back up. Take care, Seed. I hope the news isn't too bad. Yeah, take care. Mast cells and eosinophils? Oh, eosinophils also are allergic? I thought they were all only parasitic. Oh, parasitic. I'm memeing myself. I mean to say like worms. Eosinophils are both allergic and parasitic, and some cancers. I see. Yeah, I'm not too familiar with that end of the immune system. All right. Yeah. So The mechanism by which T regulatory cells work is still under intense investigation. I actually couldn't find a whole lot about how these cells work, but the fact that they exist is worthy of a Nobel Prize. So, today we're specifically going to look at the contribution of one of the Nobel Prize winners, Shimon Sakaguchi, who was the first to identify the T regulatory cells. So I, just didn't have the time to cover the other two people's work. So I really just wanted to do a deep dive as to what the first paper to really identify the nature of the regulatory T cell was, or to be able to actually isolate them. And unfortunately, if you're a part of my Discord, you already know this. Old papers are almost always copyrighted, which is super annoying. So I won't be able to show you actual data, which is a bummer because the data is cool. So you'll just have to deal with my PowerPoint presentation instead. All three of them got the Nobel Prize? Yep. Sakaguchi was the first, but not the first to fully flesh out the idea and existence of these regulatory Ts. So we'll go ahead and take a look at the discovery made in this historic paper published in 1995, Immunologic Self-Tolerance Maintained by Activated T Cells Expressing IL-2 Receptor Alpha Chains or CD25. Breakdown of a single mechanism of self-tolerance causes various autoimmune diseases by Sakaguchi et al. So of course, as per usual with these biology papers, the jargon in the title is going to be pretty hard to parse through, but don't worry, we will walk through this discovery together. But before we begin at the beginning, we actually have to go back all the way to like 1969 or something, because we can't actually talk about this and have you understand the insight that was required to get here without taking a look at the existing literature. So let's go ahead and time travel back to the 1990s. And I know, according to my analytics, there is a portion of my audience who this will not apply to, but for the rest of you, let's go ahead and time travel to the 1990s. The Game Boy came out. Pokemon was, becoming a thing. GeoCities, that's cool. VHS tapes were around. Y2K was concerning. And this was all before a time when we really understood the nature of autoimmune diseases. Now, at this time, we actually do understand that autoimmune diseases exist, right? We know that autoimmunity happens, but we don't really know why. In fact, we have no idea why or how autoimmunity happens. So for a second, we have to consider that these Nobel Prize winners don't just emerge out of a vacuum. They don't just pop out. They are steeped in a rich literature that came before them. So what does the literature say about autoimmunity? So in order to kind of recreate the environment that Sakaguchi was operating in, we should think about how we would try to explain autoimmunity. And let's use this spectrum of immune response strength as a scaffold. So what are two different models by which autoimmunity / overactive immune systems can be triggered? And use the broadest language. Pretend like you never took a biology class beyond high school, and using this picture, tell me two broad general ways, don't use the language of biology, just two general models by which autoimmunity can develop, or an overreactive immune system can develop. Yeah. It really shows that we take so much knowledge for granted when these people had to work in a figurative cave with a box of scraps. The thing is that, I'm sure in 20 years, people will look back to our time and also believe that we are working with a box of scraps, right? And that's why I think it's useful to imagine, ourselves in their shoes, because even though we know a lot more, to practice thinking with limited tools is going to lead to the next Nobel Prize innovation, right? Because you are always limited by the tools you have at hand, and the real innovative insight is always going to come from making the best out of your tools. So it's 1990, you don't know how autoimmunity works. What are two general models? Autoimmunity is sometimes epigenetic. Yeah, we don't really know a whole lot about the genome. This is before the Human Genome Project. So epigenetics, you could theorize about it, but the tools to really dig into epigenetics in the human scale is not really there yet. So two different ways. You can figure it out just by using this diagram, right? How do you get to the right hand side? Infection can trigger it. Avoid biology language. Okay, we're thinking general concepts here. And the reason why I want to start here is because a lot of, if you start too specific, you end up missing the forest for the trees. And I'm trying to help train a structured, ordered way of thinking so that you don't miss things by only seeing the trees when you could be seeing the forest. Immune system picks up on the wrong detritus at the site of infection, or it picks up the correct detritus, but it's close to something else that is not detritus. Again, too specific. Let's go very, very broad. Very, very broad. Very, very broad. How come not here, not there? I saw many studies, sure, but were they from the 90s? From the 90s. And also, epigenetics is a bit difficult to fully understand because the impact that epigenetic has on an actual biological mechanism is a little correlative, right? It would take a bit more work to connect epigenetics to anything solidly biology, if that makes sense. So let me sort of prompt you a bit more then, because I think the fishing that I'm trying to do maybe isn't working so much. How can that arrow that describes a perfect immune response be moved to the right? There are two different ways, or actually there are three different ways that that arrow can move to the right, but I'm looking for two of them. How do you move that arrow? How do you move that arrow? Ignore biology for a second. Ignore biology for a second. How do you move that arrow to the right? No. We're banning the words immune system. We're banning the word biology. We're banning the word cell. Just tell me how to move that arrow to the right-hand side. Push it. Okay. Yes. Sierra Smith, you got it. You can push the arrow to the right. How else can that arrow be moved to the right? You can use Photoshop. True. You could use Photoshop. So you can push it. That's definitely true. What's the other way? Pull it. You can definitely pull it. That's actually the third way. Or a loss of regulation, as Trojan had said. In other words, right? You can push it, right? There's something driving autoimmunity, which increases reactivity. So this arrow can be pushed to the right. And then Mr. Ogre says no stopping. Yeah. So what if this arrow is dragged to the right, but then there's something stopping it? And when you remove the wall, then it gets pushed to the right, right? There are two models here, right? Two models. Something that pushes the arrow to too strong of a reaction, or you lose something that was holding this arrow back and then it gets dragged to being too strong. Does that kind of make sense, chat? Does that idea make sense? And again, I'm not saying you are all wrong with the stuff that you were saying, but in terms of like developing abstract thinking, which is really, really important for when you're trying to do innovative stuff, right? If you are bashing your head against a problem that other people have failed to solve, sometimes you need to think very simply and abstractly, because maybe other people are just getting lost in the forest. They're like too deep into it and can't return to a state of simplicity. Because if you start from a simple idea, then you can explore all of the different options instead of being stuck in a particular minimum, right? So this is what I mean, right? You can either be pushed to the right or you can have the block removed. And I know that might seem okay, like why am I learning it this way? Or why am I insistent that we think about this silly, stupid example? It is because when you are engineering, you should think in simple terms, because then you can explore multiple different options. Okay, so really wanted to drive home a simple explanation first before we get into the specifics. So maybe there's something stopping autoimmunity, and when it's removed, autoimmunity happens, right? And this is the insight, right? This is the model by which Sakaguchi and people who agreed with Sakaguchi are assuming. Because again, the reason why Sakaguchi won the award and not some other person is because Sakaguchi took a chance on this more obvious scenario, this pushing scenario, not being true, right? Sakaguchi believed in this scenario, this a little less intuitive, but still very simple to conceive of opportunity, right? I'm new to this channel. This is a biochem channel. It's a molecular biology and cell biology channel. I think that's the most accurate way to describe me. I think oversimplification is an important exercise for development. Exactly. Like oversimplification, you start there first so that your thinking can be clear and straightforward. Okay. So now we can talk about the actual background. Now that we have the theoretical framework, right? People back in the 90s, no idea how autoimmune reactions happen. There were two hypotheses. One, that autoimmunity is driven by something, or that autoimmunity is caused by a loss of regulation. These are not mutually exclusive. They can be both, right? They can be both. But, how do you decide which angle to tackle? Sakaguchi wanted to tackle the idea that this could be a loss of regulation. Okay, so we have to go back all the way to 1969 for Sakaguchi's first kind of theorizing about this system, the regulatory T cell. And so these other Japanese scientists, Nishizuka and Sakakura, found that if you remove a mouse's thymus, where T cells mature, that also weirdly caused destruction of their ovaries. It was later discovered that those mice's ovaries had disappeared because the immune system destroyed their ovaries. And that's wild. I bet they were pretty surprised. I have no idea why Nishizuka and Sakakura were removing mice thymuses. I think a lot, back then there was a lot more basic biology that was being done, like what does the thymus do? And then you remove the thymus and see what happens in the mouse. And unexpectedly, by removing the thymus, the ovaries also disappeared. Did they know that T cells matured in the thymus? I'm not actually sure what they knew about the thymus back then. Why would we talk about autoimmunity as a single concept? As with cancer, there are many things that may cause it. That is true. But in terms of a key central figure in the story of autoimmunity, this discovery is paramount, right? As I said before, like autoimmunity is not just this one thing, but this one thing does contribute a lot to it. If you remove a mouse's thymus, sounds like if you give a mouse a cookie. I'm sure the mice that got the cookie is going to be much happier than the mouse that lost its thymus. So this is a neat hint as to what the mechanism behind autoimmunity could be, or at least some versions of autoimmunity could be, right? So you remove that thymus and all of a sudden the ovaries disappear. So the thymus, probably pretty important for making sure that the body doesn't destroy itself. And so just a couple years later, there's this Penn-Hale hypothesis that hypothesized the existence of a suppressor T cell. But you can imagine with the driver model, people were reluctant to speculate on the existence of yet another cell type, right? Like, Occam's razor, right? The most simple explanation is often the most correct, right? And so in that diagram I had showed, it's easier to believe that you just push the arrow to the right and that's what causes autoimmunity. And so people were not readily willing to put their money in the idea that there could also be an immune cell that is really an anti-immune cell. That's maybe more why people might not have really thought that this was possible, because the immune cells all gung-ho destroying things. How could there be an immune cell that is anti-immunity? Seems kind of weird. Of course, now it seems pretty easy to believe, but back then it was a lot harder of an idea to swallow it. Okay, so in 1974, it was discovered that mice have autoreactive B cells against their own circulating blood. And this was also pretty interesting as well, right? So this is, of course, another old copyrighted paper that I can't show, so that's annoying. But what they discovered was that healthy mice, if you isolate their, their sera, you can find autoreactive B cell antibodies against their own circulating blood. And that's pretty interesting as well, right? There are many red blood cells, right? Your immune system comes into contact with red blood cells all of the time, right? And so it comes to reason that maybe, you would maybe develop an autoimmune reaction to your own blood. And yet healthy mice do not suffer from autoimmune disease, right? And you can kind of see how these findings are shaping Sakaguchi's mindset, right? Is the immune system being held back from autoreacting, or is it being driven to autoreact? And again, Both models are totally valid for different autoimmune disorders, but we're talking from the perspective of a healthy mouse here, a healthy mouse. Even a healthy mouse is developing antibodies against their own blood. But why doesn't a healthy mouse develop autoimmune disease, right? So I'm going to ask you this question. You discover this paper from 1974, the fact that healthy mice develop antibodies against their own blood. And now, does this finding convince you or lead you to believe that autoimmunity is driven primarily in healthy mice, or mice starting from a place of health, or something that is no longer held back by something? What would you say? What would you say? The finding that healthy mice, even healthy mice, develop antibodies against their own blood. Another sip of my I'm just having hot water today. And that's weird. It is weird, right? And it would lead you to believe one particular model is at least at play, right? At least at play. No longer held back makes sense to me. Held back, they are no longer held back. Yeah, right? Because your body makes a lot of blood, right? I don't remember how much blood, 2 liters or something. I don't remember how much blood is circulating in the body. It's probably more than 2 liters. For certain, more than 2 liters. I don't know why I even tried to name a number. But yeah, your immune system is in contact with blood all of the freaking time and is developing autoimmune responses to that blood, but you don't develop an autoimmune disease. And that seems to imply that the immune system is being held back by something. And so, people in Sakaguchi's camp are concluding that something is suppressing an autoimmune response, that it is not solely driven by something, that there is an anti-immune immune cell, which again is kind of hard to accept at first. So, oh my gosh, maybe I shouldn't be streaming at this hour while there's machinery work. Let me go ahead and reposition my microphone a little further from the window, and maybe that'll help the problem a bit more. In context, that makes sense why it's held back. Your body is exposed to self-antigens continuously, and that is what Cunningham et al. Had concluded. Certain facts, this is a direct quote, certain facts suggest that this constant level of plaque-forming cells, this is just a broad term. This is language specific to the test that they were doing. Plaque forming cells being cells that are producing an autoimmune response, right? So PFC, if you see PFC, is just in this test, these cells are making autoimmunity happening. So certain facts suggest that this constant level of autoimmune reaction in normal mice is maintained as a balance between two opposing forces: continual antigenic stimulation by self-antigens on one hand, so driven partially, and some suppressor mechanism on the other. Constant stimulation seems likely since the antigens form part of the normal mouse red blood cells and probably become exposed as red blood cells degenerate. So when red blood cells die, they spill their internal guts everywhere, and those proteins are going to elicit an immune response. And so because you don't develop immunity to your own blood, at least most people don't develop immunity to their own blood, it implies the existence of a counterbalance, right? Not only is immunity driven, but it is also counterbalanced by something. So cells could either be taught to attack bad things or taught to not attack good things. Exactly. That's a pretty good way to think about it or to conceptualize it. With this paper, we will learn that it stands to reason that the model of not attacking good things has some evidence to support it. Exactly. Will this live stream be available afterwards as a VOD? All YouTube VODs are automatically archived, so you can find it in my live tab on my channel. Wait, is this paper by the same guys who won the Nobel Prize? No, this is by Cunningham et al. So Sakaguchi had nothing to do with this paper. In fact, this preceded Sakaguchi's work by at least 10 years in terms of when Sakaguchi was doing science. So you can see that, Nobel Prize winners don't just come from nowhere. You see this often in media that like Nobel Prize winners have this eureka moment, and this idea was given to them by a higher being, divinely inspired, but it's not actually true. You can see the building blocks that were helping Sakaguchi come to the conclusions that he will conclude to. Right. So it's surprising, right? Because this already determines, Sakaguchi didn't discover the mechanism of autoimmunity. Sakaguchi discovered the cell that was responsible for a lot of suppression of autoimmunity, but that had to have come from a research milieu that justified his existence. Okay. All right, so you be the scientist. Here again, I promised at the beginning of stream that I would be very interactive because I want to show you that Nobel Prize winners are brilliant, but their brains are not doing anything that your brain can't, right? That is two different concepts that you must hold together. That Nobel Prize winners are still human, but they are very, very brilliant. And we're going to help you walk in their brilliant shoes, right? So here are some things that we understand about autoimmunity. Autoimmunity is theoretically held back by something that breaks in autoimmune disease, right? When you have a disease, we can speculate here, it's not concrete at this moment, but if you remove the thymus, the mouse's ovaries get destroyed. So there's something broken in the biology of autoimmune disorders, right? So what experiments would you run to find out what that something is? What would you think to do? And I'm not going to really, I'm not fishing for a correct answer, okay? I just want us to take this meditative time to attempt to be a Nobel Prize winner, right? Just attempt. And the reason why I do this on stream, if you've never attended stream before, is because, I don't care if you get the right answer. In fact, if you've read this press release, you already know what the right answer is. What's more important is that you try to think about an abstract problem. And even if you're wrong, you still forced your brain to come up with something new. And I think that is valuable as a teacher. Are we doing theoretical biology now? Kind of speculation to try to put yourself in the shoes of a Nobel Prize winner. Remove things. That's an interesting approach, right? Remove things. And I like that you said that, because that is the kind of accessible, broad language, the kind of oversimplified language I'm looking for. We don't have to get in the weeds with the Nobel Prize winner to be able to appreciate their brilliance, but we should try to kind of speculate and theorize in exactly that sort of language, right? Remove things. Now we can get more specific. What do you want to remove? What do you want to remove? Nobel Prize winners are people that have research equipment and money, and we don't. That is true, and they also have drive and spirit, and they are steeped in the research. But again, their brains don't actually do anything different than yours. It's just they are fiercely devout and devoted to their craft. Compare the blood between a normal person and someone without a thymus. That I don't know if that would work, because I don't know how many humans have survived without a thymus. I mean, if there are if there are conditions where humans can lack a thymus and survive, that would be interesting. That would be interesting to mine more information. It says remove specific types of white blood cells. Yeah, removing stuff would be good too. Removing stuff. I would try to break that something, take immune cells and expose them to a common cause of some autoimmune disorders and see what happens. Also an interesting and excellent idea. Add some locking to debug. Yeah, I mean, if theoretically speaking, sure. Remove things that don't have known functions. Even to do that now, that would be a lot of work. Can you imagine how little biology we knew in 1990? I would say that that one's going to be a bit more on the impractical side. Compare what blood cells do not appear in blood in people with autoimmune disease. That is not a bad idea as well. However, there are some correlates that would make that somewhat difficult, right? The expression of autoimmune disease can be quite varied, and there might not be one common way. I'm not saying that's wrong. I'm saying that might be a little messy because some autoimmune diseases have different mechanisms than others. So it's certainly a good idea, to refine that process. Yeah. All really good ideas. Comparing blood samples is a really, really good idea. And, in terms of gathering more correlates to try to hone in on the background, I would say that is correct. However, Sakaguchi is out for blood, not the blood that you're comparing, but Sakaguchi is out to discover the thing. So Sakaguchi is going to take a bit more of an experimental approach, which I think is interesting. I'm surprised that amongst all the people that are suggesting things, that we haven't heard about this, because a common answer when I ask these questions is mutations. And the challenge mode for this, which you all have unwillingly, like unknowingly, kind of abided by this challenge rule, is that you cannot do mutations at this point, right? In 1990, high throughput screening in mice is $1 gajillion, right? We don't have CRISPR back then. So this was pre-modern gene editing methods. Not saying that there weren't gene editing tools back then, but CRISPR just makes it so easy and so fast that in order to do a mutation in a mouse at this point, you have to have a really, really good reason. And if you're kind of bumbling around, not knowing what you're trying to find, making a mouse is a huge waste of money back then. Is there a way to do cell count based on CD type? Oh, interesting. Keep that in mind. Keep that in mind. All right. I'd increase the amount of reactive oxygen species and induced nutritional deficiencies, which would lead to functional dysfunction. But how do you know? So trying to elicit autoimmunity by general cell damage, I'm not entirely sure. I mean, it would work, but I'm not entirely sure what you would get out of that, right? I'm not sure what information you could recover. Okay. We would also need to find a target gene and a target cell, and I think that would take a lot of work. Exactly. That would be quite difficult because genetics at this point is pretty rudimentary to the tools that we have right now. Smack different types of T cells with antibodies and see if there's a cell type that makes autoimmunity worse when disabled. Ooh, keep that in mind. Think about that. Interesting. All right, so we have a bunch of good ideas out here. And I kind of forgot that I had this bullet point, so we'll just move on, right? So we have a couple of different approaches, a lot of which I think are very valid, right? You can continue to mine correlative data and try to compare patients with autoimmune disease to patients without, and try to approach it from that direction. I think that's valid. I think it can be a little messy because biology in a human is messy when you don't have experimental tools. So I'm not saying it's bad, but I'm saying that's not the route that was taken, which again, I think is totally valid. I think it's cool. And maybe in an alternate universe, that is the route that Sakaguchi would have taken, but that's just not how reality shook out. So I like that idea of comparing blood. I actually didn't think about that myself, is to compare blood in that way. And I think that would be neat. No mutations and drugs/medications that act on the immune system. Some help against autoimmunity, but were they known at the time? But then what would you do from there, right? At the end of the day, we want to think about trying to find the thing that is causing autoimmunity. And there has to be a recoverable piece of information that you could get from these experiments. I have a terrible idea. Take the white blood cells from a biologically identical mouse and just put it in another mouse that doesn't have a thymus, if that's possible. Oh, I mean, not a terrible idea, right? Not a terrible idea. Grafting is a biological technique that is used in the story, although I don't know if I have it in these slides. Okay, so the tool that was used a lot in the build-up to Sakaguchi's paper was a tool called anti-lymphocyte serum. So this was something that chat has suggested, is to just try and kill different things that come out of the thymus and see if you develop autoimmunity, right? So you can make a serum that kills T cells. And you do this by injecting an animal like a rabbit or a goat or a mouse with human lymphocyte proteins. So you give them a cocktail of human T cell proteins, and those animals will develop antibodies. And remember, they have an immune response too. So they'll develop antibodies to your T cells. And then you can take that anti-lymphocyte serum and destroy specifically human T and B cells. So this is a technology that is not only a useful research tool, but is also useful for graft-host disease, right? So to make sure that when you get an organ transplant, that your immune system doesn't kill the organ, right? But it's also broadly immunosuppressive, so it reduces the response to bacterial antigens as well. A very, very sledgehammer kind of tool that's used to suppress the human immune response. But in this particular case, they were trying to figure out what the nature of autoimmunity was, so this is what they were trying to do. So instead of removing the thymus, you can now keep the thymus intact but just kill the cells instead of removing a whole organ, which of course would make the experiment a bit cleaner here. All right, so what do you think happens to an autoimmune response when injected with anti-lymphocyte serum? And so here is some other responses, right? So if you inject transplant recipient with anti-lymphocytes treatment, you find that the transplanted organ is no longer attacked, right? You can keep that organ safe. You have an immunosuppressant response. If you use bacterial antigens, so if you inject a mouse with bacterial proteins that they will mount an immune response to, if you inject them with anti-lymphocyte treatment, then those bacterial antigens no longer provide that intense immune response. What do you think happens to a mouse when you inject them with anti-lymphocyte treatment? What happens to autoimmunity, do you think? What happens to autoimmunity when you inject a mouse with this T and B cell killing antibody? Goes down. What goes down? It gets better. They get better. Cure. So this is specifically, hold on. I guess I worded the slide in a bit of a poor way. So let me be a little more careful with how I do this. Yeah. So say, yeah. Okay. Sorry. I had the slide a little wrong. Say you have a mouse that has a bit of autoimmunity. If you inject them with anti-lymphocyte treatment, do you expect their autoimmune condition to get worse or to get better? Think about that for a second. Okay, so we have some hypotheses. Autoimmunity increased. There was a heavier response to self-blood antigens, which is kind of counterintuitive, right? Because this anti-lymphocyte serum is broadly immunosuppressive, right? You give it to someone who had an organ transplant and their immune system is dampened. You give it to a mouse that has been challenged by bacteria and their immune system is dampened. But when you give it to an autoimmune mouse, their immune response goes up, right? They get more reactive, and that's kind of counterintuitive, right? Just stare at this diagram for a little bit. And that's interesting, right? And it's counterintuitive because in the other two situations, you had the anti-lymphocyte treatment dampen the immune response, but here in an autoimmune response, in an autoimmune mouse, the result is the opposite, right? It is increased. It's the opposite effect. Could you repeat the question? My mind got nuked reading the chat. Yeah. What do you expect in an autoimmune response? In an autoimmune mouse, if you give them a serum that kills their T and B cells, what do you expect their response would be? Yeah. Chat, you're putting it together. I can see it, right? This only makes sense if the ALS treatment, the anti-lymphocyte serum, this only makes sense if you destroyed the thing that was holding back autoimmunity. The serum removes the brakes. Thank you. That's an excellent way of putting it, right? I like, and I highly encourage chat to do this. Think in those abstract ways, in those metaphorical, concrete, not concrete, but those metaphorical ways, because that is going to help you become a better thinker when you can think abstractly, right? If I just dumped data on you, it would take a little bit of work to decipher them. But if you come in at the data from the perspective of someone who is just trying to understand the fundamental function of these things, you can start to see connections that you might not otherwise have had. Okay, so that's cool, isn't it? Right? This treatment for an autoimmune mouse does the exact opposite thing than it would normally do when you introduce other things, and I think you all have the idea down. Okay, so why does this treatment work? Is because the anti-lymphocyte serum dunks on all T cells, right? So it kills the combatant cells, it kills the helper T cells, it kills the memory T cells, but it also kills these memory T cells that are responsible for self-tolerance, right? This is, I call it a mystery T cell because they don't know at this point that this thing even exists, right? They hypothesize that it exists, right? They look at the data and they say, there must be some cell out there that is suppressing the immune response, right? There must be. Surely there must be. That's the only way this data makes sense, but they've not been able to prove it, right? Sakaguchi is the first one to show that this cell exists. and point to it, right? Isolate them, say that this is what the suppressor is. Everyone else before him was just, surely this thing exists. It's kind of like the Higgs boson, right? A lot of people have hypothesized of the existence of the Higgs boson, right? But the real meat on the bones is discovering whether or not the Higgs boson actually exists, or to measure it in some way. And you can think of it similarly here. Okay, yeah, so if you destroy these mystery cells, you no longer get that self-tolerance. Okay, so this anti-lymphocyte serum aggravates autoimmunity, potentially by removing what they called in 1974 suppressor cells. They've been renamed to regulatory T cells, but I prefer suppressor cells just because it sounds cooler. Anti-lymphocyte serum reduces the don't hurt yourself element even more. Exactly. So that memory retention is linked to lymphocytes rather than any other white blood cell. Exactly, right? You removed the thymus, and that, in 1969, removing the thymus killed the ovaries. So you think, okay, it's probably T or B cells, but the thymus is probably responsible for a whole bunch of other things, so you can't quite be sure. The anti-lymphocyte serum is a very targeted, very specific way to destroy only T and B cells. So you know that whatever is responsible for autoimmunity is either a T or a B cell. And back then, I don't really know why people thought T over B. I didn't go that deep into the literature, but people were thinking that it was probably a T cell for some reason. So if you give this serum to a healthy mouse, does it get autoimmune disease? I believe that's what they had found, although I'm not 100% sure just because I don't have the citation on screen. So Cunningham had said, the simplest explanation for these results is that the anti-lymphocyte serum promotes the autoantibody response by removing suppressor cells. The suppressors may be T cells, although the evidence is not conclusive. So I want to exercise caution here and teach you all a little bit about biological skepticism, right? Or healthy skepticism. So Cunningham, even though I think this is pretty slam dunk, right? This is pretty slam dunk, but Cunningham is very careful, and I think responsibly careful. So why is Cunningham careful? So here's the data that we have. Anti-lymphocyte serum, you give that to a mouse, and that causes T cell death, and you get an antibody response. This is true, right? You can measure T cell death as a response to ALS, and then they get an an auto-antibody response. But why is Cunningham saying that the evidence is not conclusive when I'm saying this looks pretty much like a slam dunk here? So why is Cunningham so careful? Why is Cunningham so careful? Maybe because B cells need T cells to get activated. That's true. B cells are activated by those helper T cells. So why is Cunningham saying that even though he suspects that the suppressor is a T cell, why does he suspect, or why is he not very willing to commit to that idea? Could other things have been destroyed in the making of that we haven't discovered yet? Exactly. In other words, what is this paradigm called, Ari? So Ari, you've nailed it on the head. The ALS, the anti-lymphocyte serum, could be doing something else, right? It doesn't, it definitely causes T cell death, definitely causes B cell death, but you can't say it was a B cell or T cell or something else. Yeah, exactly. What you can say is that the ALS causes an auto-antibody response. That is for sure true. You can say it's probably the T cells, but we don't actually know at this point in 1974 that the ALS serum is doing something else. Right? Maybe it's killing a T cell, and maybe that T cell is killing maybe not having that T cell is killing something else that's the actual suppressor, right? The details in between anti-lymphocyte serum and auto-antibody response are not fully fleshed out. So Cunningham does the very, very responsible thing and goes, it's probably the T cell, but maybe it's something else, right? And that just gives you an idea how careful you have to be as a scientist. Even though it sounds like a slam dunk, it actually isn't, but it's pretty close to a slam dunk. Okay, so a suppression model for autoimmunity, right? I said these two models can overlap. They're not mutually exclusive. So regular immunity drives reactivity mostly, but not exclusively, through non-self antigens, right? You get exposed to some bacteria thing, you mount an immune response, you get exposed to a cancer thing, you eliminate the cancer, et cetera, et cetera. So there's that regular immunity that is responding to proteins and attacking them. And amongst those proteins are your own proteins, right? Your blood proteins, et cetera, et cetera. And to prevent a response from mounting against your own cells, there's this other mysterious cell that is holding it back, that is tuning the immune response, making sure that it is only as active as it needs to be, that it does not go overboard and start attacking yourself as well. Okay. So this is the model, suppression model for autoimmunity, right? It's not solely driven, it is also held back. And again, that was pretty new and exciting at the time. And yeah, the idea of an anti-immune cell didn't really sit right with people, which is why these innovations are so wonderful, right? Sakaguchi took a chance on this. So by the time Sakaguchi published his 1995 paper, which is the Nobel Prize winning paper, or one of them, the existence of a suppressor T cell was becoming more and more likely, right? Sakaguchi didn't, discover the idea of a suppressor T cell. Sakaguchi was the one who actually found it. But people leading up to this were pretty sure that this thing actually existed. Wait, so at 74, wouldn't they agree on the suppression model, even if they don't agree on what's performing the suppression? I'm not too well read on all of the literature by 74. I just know that it took a while for people to accept the existence of a suppressor cell. On RBCs, basically just sacks of hemoglobin. They also have other antigens on their surfaces too, right? Like O antigen, right? A, B antigen, all that stuff as well, right? So they have unique identifiers that, you can imagine hypothetically a cell could develop a resistance to. So how would you try and find this suppressor T cell? We kind of answered this already, where people were just saying, well, you just start deleting things, and that's not a bad idea at all. In fact, that is the approach that people were trying to take, right? Trying to rule out or try to figure out when you remove specific types of T cells, what do you get? Do you get suppression when you remove this type of T cell, that type of T cell, et cetera, et cetera? Okay, so overall plan here, try and categorize T cells. Try and categorize them. Delete them in a mouse until you get autoimmune disease. This is an approach that chatters have recommended when I last asked this question, so I'm not going to ponder it all too much longer. But here's the thing, how do you identify a T cell? And I know that there are people in chat, I've read their messages before, they already know the answer here, but I'm going to ask you to not remind chat what the answer here is. But how do you identify T cells? Yeah, asking a very excellent question. How do we even tell T cells apart? And I want to take a little bit of time to ponder this question. It's an interesting question, and to be honest, as an undergraduate, I thought this was like the most boring question imaginable. But as full of a biologist as I think I'm going to get, right? Like I'm no longer actively doing research, so my growth has kind of plateaued. Although because of my YouTube channel, I still read almost every day, reading some biology paper. But like I think I have reached maybe 80% of my ability to do biology, and maybe I'll get a little bit better in the future. But as like an 80% realized biologist, this question is a lot more interesting than I ever gave it credit for. Do T cells have specific shape or express specific surface proteins? Interesting. I have no idea how to. My level of academia and biology was the MCAT and being a hobbyist. Oh, well, I mean, if you had MCAT biology, you might have the tools to figure this out. Membrane receptors, interesting. Expressed proteins, yeah. So, what do you think? Let's take advantage of being spoiled in 2025 and do single-cell RNA-seq. Nah, I can't do that. There's something beautiful about old-school biology where we don't have these OP technologies that let you just click a button and do it. This was old school. In fact, this isn't, even though I say this is old school, it's not that old school. This is a technique that is still done today. It's still very, very, very useful here. Okay, the people who know biology showed up. CD antigens, clusters of differentiation, proteins, etc., etc. Yes, yes, yes. That's the actual answer. But I'm glad that we got to pressure some listeners into thinking about this really interesting question, right? Do they have differences that we can discriminate against? And this is kind of a philosophical question, right? There are certain cell types where you can very easily just look at them under a microscope and be like, yep, this over here, that's a neuron. This here's a T cell. But what happens when the differences between cell types are not all that clear, right? What about cells that look identical to one another, right? This is a T cell, but certain cells have unique morphology. T cells all just kind of look like this, as Guy had questioned, right? Are there actual morphological differences that we can discriminate against? And the answer is that for certain cell types, that's going to be harder, right? All T cells kind of just look the same. Plus, a visual read on an image is not exactly that you can filter cells by, right? So not only do you need a way to discriminate between cells, but you need a hook, a handle, a way to actually isolate and purify a type of cell. And just looking at them is not going to do it, at least not with your eyes, or a light microscope, at least, as some people are hinting the use of fluorescence might be kind of useful here. And I think that you are correct. All right. So, to differentiate between T cells, if they look the same externally, they might look different internally based on the way they store different proteins. Definitely true. Not the approach that they take, Frost. They're still going to be looking at the outside, but they're not going to look at them with a light microscope, right? A microscope that just gives you the shape. They're going to be a little clever about how they look on the outside here. So how else can you determine cell type? And, you know, I've had chatter spoil the answer already, but that's totally fine. No worries here, no worries. Biology is, and I'm wondering if we have chatters here who have listened to me talk about this for long enough, who can help me fill in this blank? This thing that I say in, I think, every 5 video. I talk about this. A bunch of shapes interacting with shapes. Exactly. Biology is just a bunch of weird shapes bumping into one another, right? And if we zoom out there, it's also a bunch of bags with a unique collection of proteins bumping into one another, right? So this is a corollary. It's not the thing that I normally say, but it is a thing that you can conclude, right? If cell biology is weird shapes bumping into one another internally within the cell, then, you know, if you zoom out, then all, you know, tissue biology is cells bumping into one another, and cells are coated in these weird proteins, right? So here is the thing. Because we know that cells have weird shaped proteins with unique conformations on their outside, we can actually use those proteins as a handle to sort them by type. You said that during the stream twice before. I you got me. You totally got me. Can you tell I don't spend that much time on these presentations? Don't get me wrong. I put in a lot of effort into these, but I leave them at about 70% perfect is what I would think to myself. Yeah, so because cells are coated in these proteins, we can take that fact to our advantage, right? And every cell has a unique combination of different amounts of proteins that we can then sort them by. So if we go back to this philosophical question of what even is a cell type, well, you know, as Ari had pointed out, biology is also a spectrum. Biology is also a spectrum, right? I have 2 things that I say about biology. It's just a bunch of jiggling shapes bumping into one another, and then Ari's got the second part. Biology is also a spectrum. So if we want to ask what the philosophical definition of a cell type is, unfortunately, as I have said multiple times on this channel before, categories and labels are just not that useful, right? I mean, they are useful, but they're not perfect. Cells are typed by the identity, quantity, and combination of proteins that they make and house. So Frost had mentioned that proteins within the cell, yeah, proteins that they make inside the cell also definitely count, but the outside cells are easier to handle. Inside cells, you know, those things are a little harder to interact with experimentally. If I just got back from a hunting trip with my son and decided to watch some biology to relax, thank you for choosing me as your post-trip entertainment. Very, very cool. What is biology? It depends. Exactly. People who simplify biology into strict categories and labels think that one word means the same thing in every given circumstance. They're just not doing biology right. So, cells that make a lot of immune proteins, for example, are probably going to be immune cells. And note that I say probably, right? Like labels are useful because they help you describe a bunch of disparate data, but labels are not concrete barriers, okay? Labels are fuzzy, okay? Especially in biology, labels are very fuzzy. So each cell type has a unique protein fingerprint that we can attempt to classify them by, right? This is a very important concept, right? Even though you can say this is a neuron, and a neuron is definitely different than a cardiomyocyte, right? But neuron and cardiomyocyte, those are two cells that exist on a sort of spectrum, right? They're both electrically reactive cells that share a lot of similarities, and we just call things that are neuron-like neurons, and then we call things that are cardiomyocytes cardiomyocyte-likes, right? This is a very important concept in biology, that these labels are not that concrete. So, hopefully, even though I've been crapping on the idea of labels, I'm only saying that because people get too invested in labels. When they think that one word is one word forever and it doesn't mean anything else, that's just not true. But that doesn't mean that they're useless, right? The label of a neuron is still very, very helpful. It's just, you have to acknowledge that when you say neuron, that the boundary of the word neuron is very fuzzy. So hopefully, if you're lucky, you can find one unique protein that can act as a marker. We call these cell markers, right? So for neurons, this is very simple. There are neuron-only proteins that when you try to throw a fluorescent antibody against that neuron-only protein, you can identify neurons. So hopefully you can find one for every cell type, although that doesn't appear to always be the case, annoyingly enough. But how can we be sure that the protein is unique to that cell? Excellent question. Just more testing. You just have to do more testing. You just have to do a lot of testing to see whether or not that protein marker is expressed anywhere else and in what levels. Okay. So, where you choose to draw those borders between what are cell types is debatable because, again, labels are always fuzzy at the borders. But, if we can all agree on a vague boundary, then that is very helpful because we get to communicate science that way. So who is going to be the first one to identify the suppressor T cell by their cell type, right? This is what the search is for and why the Nobel Prize was given to Sakaguchi. He found the T cell, suppressor T cell marker, the identity. Okay, so a couple of chatters have teased the existence of these proteins, the CD proteins. If you've taken any amount of immunology, this is going to come up time and time again. These CD stands for cluster of differentiation. This is a globally agreed upon nomenclature for proteins on the outside of immune cells, helps them to identify the difference between a T cell, a B cell, a monocyte, etc., etc. So when you see CD, you are often referring to an immune marker, although, beyond that, they don't necessarily have similar functionality, right? In fact, all of these CD proteins do something unique. It's just that when immunologists were trying to figure out immunity, they needed a way to keep everything straight. And so they developed this pretty horrible nomenclature, in my opinion. So there's this existence of these CD markers, and you'll see that from the stem cell, if you are going to be destined to be some sort of lymphocyte, some sort of immune cell, that stem cells that differentiate and inherit a CD45 protein, you can see this on top here, they're probably going to be some immune cell of some sort. Although you can see thrombocyte, I don't remember if thrombocytes count as an immune cell, but you can see that most things that don't have CD45 probably, or at least in this unique combination, probably not going to be immune cells, right? So if you have CD45 and CD61, you're probably a thrombocyte. If you have CD45 and CD19, you're probably a B cell. If you have CD45 and CD3, you're probably a T cell. If you have CD45 and CD14, you're probably a monocyte. 45 and 15, granulocyte, right? So I was drawn to the concept when trying to learn about embryology tissue differentiation. Super interesting, right? The idea of cell destiny is such a romantic idea, right? The destiny of a stem cell. And so the destiny of a stem cell can be tracked by looking at what proteins they are making. I don't get it. If there are 4 types of T cells, why was one significantly harder to identify than the other 3? Which one was it getting mixed with? That's because I just think that it took more time for this one. I'm not like a T cell expert in terms of like the history of these discoveries, but I do know that there were a couple of failed attempts, and I do think that the phenotype of autoimmunity is a bit harder to get straight given how it is done, right? Because, bacterial challenge, you just inject a mouse with bacteria and then you can study regular immunity that way. Autoimmunity is kind of trickier in certain ways. Why are B cells not here? They are. B lymphocyte, right? Up here. Or is it a single T cell that specializes into behavior in certain circumstances? Yeah, so this T cell, this CD3 positive, CD45 positive T cell, this differentiates into a couple of different cells: the helper T cell, the cytotoxic T cell, and the activated T lymphocyte. Or when it is activated, it expresses CD25, right? So that's just the general idea behind these CD proteins, that they are surface cell markers that are especially useful for identifying immune cells. Why do they all have CD45? Is it a protein that does very important things? I'm sure it does. I actually don't know the answer to that. It's an excellent question. Again, yeah, I'm not like an expert in T cell biology. So lymphocytes are classified by their surface proteins, right? Most of these proteins are called CD blank for cluster of differentiation. It's just a shorthand for immunologists to try to keep their story straight with regards to differentiating cells. So again, cytotoxic T cells, they make CD8. So when you see CD8 positive T cells, you think killer T cells. These go by a couple of different names. Helper T cells make CD4. And then, so the question that becomes, what does the suppressor make? What does the suppressor make? So Sakaguchi was actually circling this target as early as 1985, right? Sakaguchi took a stab at trying to find the cell marker, but failed in 1985. But we'll see that his failure to identify the T cell 10 years before wasn't really a failure. It was just a stepping stone to the Nobel Prize. And so this paper is called Organ-Specific Autoimmune Diseases Induced in Mice by Elimination of a T Cell Subset: Evidence for the Active Participation of T Cells in Natural Self-Tolerance, a Deficit of a T cell subset as a possible cause of autoimmune disease. So before this, we were thinking, you give the mouse the ALS, the anti-lymphocyte serum, and it gives them autoimmune disorder. But we're not sure if it's a T cell, we're not sure if it's a B cell, we're not sure if it's something else. Sakaguchi in this paper shows that it is in fact a T cell of some sort, not just a weird effect of the serum. Okay. So, what did they find in this paper? So Sakaguchi took a stab here, right? Sakaguchi tried and eliminated, or rather, Sakaguchi knew that So Sakaguchi took inspiration from that 1969 paper, right? That paper that removed the thymus and then that caused the ovaries to disappear, right? When you remove the thymus, you remove these T and B cells. That causes the immune system to attack the ovaries. So Sakaguchi went back to that paper and thought, okay, what can we learn about those mice that had neonatal thymectomy, right? Taking the thymus out of a mouse. And so what Sakaguchi had found was that when you removed that thymus after the mouse, neonatal, so shortly after the mouse is born, that there's a huge drop in the CD5 T cells that exist. Big drop in CD5. So Sakaguchi thinks, okay, let's just try CD5 here. Let's just try CD5. I mean, it could be some other T cell, right? Like removing the thymus is removing a lot of T cell differentiation stuff. But he noticed that CD5 was especially important for some reason. So he raised serum against CD5 specifically. Remember that you can get this neat trick where you can assassinate specific subpopulations of cells just by injecting an animal with them and causing them to get an immune reaction, right? This is, by the way, how antibodies are made, right? In terms of if you have an antibody treatment for a disease or as a research tool, you often have to inject an animal, get them to make an antibody for you, and then you can use that antibody. And so he took CD5, human CD5, or rather, sorry, he's doing this experiment in mice. So he's taking mouse CD5, injecting it into some other animal, I think a rabbit or something, or a goat, and then harvesting their antibodies and using that to kill specifically the CD5 positive T cells in the mice. So, autoimmune disease results. That's interesting, isn't it? So, instead of using the anti-lymphocyte serum, this broad sledgehammer where you're killing everything, T cells, B cells, and then causing autoimmunity that way, Sakaguchi is taking a surgical approach, only eliminating the CD5 T cells by using this anti-sera approach. Right? That is interesting. So what happens? He found that, yeah, autoimmune disease happens, but CD5 is like not a great marker here because plenty of other cell types make CD5 as well. So, you know, that's kind of a bummer. You know, Sakaguchi finds that this is a T cell phenomenon, right? He killed the CD5 positive T cells, but other T cells are also CD5. CD5 does not give you the correct category. Okay, but he did show that it is T cells, right? Because his CD5 anti-sera does not affect B cells and it doesn't kill the thymus. So he shows here that it is in fact some kind of T cell that is suppressing autoimmunity. But CD5 is just not it. It's not a great marker because other T cells make this, which is unfortunate. Very, very unfortunate. So, you know, I bet Sakaguchi was pretty bummed here, right? Because, you know, he's taking shots at this target. Like, he wants to find the thing that will allow him to find the T cell that is suppressing autoimmunity. And he gets closer and closer and closer. He like latches onto this problem and doesn't give up until he finds it, which is pretty inspiring. So, another group had found that elimination of cells lacking CD45 also caused autoimmune disease. So Sakaguchi is looking at other literature, seeing what other people are doing. So even though his CD5 result was a bust, there's another group that found something else that looked very tempting. But again, CD45, lacking CD45 is also not a very specific marker, right? So eliminating CD5 cells and eliminating CD45 cells were both correlated to autoimmunity. They both cause autoimmunity in a mouse, but neither is good enough to be the criteria that we identify the suppressor T cell on. So now I want to sort of turn this back on you, Chad. And this is the insight that I think wins Sakaguchi the Nobel Prize. So here it is. What do you think Sakaguchi did with this information, right? And if you've just joined us, I'll just give you a recap. Sakaguchi is trying to find the cell that causes suppression of the autoimmune response, the cell that protects the body against attacking itself. He tried and did find that when you assassinate T cells that only have, or when you assassinate T cells that make a lot of CD5, you get an autoimmune response, but CD5 is not very specific. Another group found that T cells that were not expressing CD45 would have a relatively low level of this protein. If you eliminate them, they also give you autoimmunity. So what do you think Sakaguchi did with this combination of information? And this is the key Nobel winning insight, at least from my perspective. Any Nobel Prize winners in chat want to put these two findings together? It's very odd fight to have serum kill a very specific CD is something I can't even picture. Well, you can imagine if an antibody is against CD5 specifically, then when you inject that anti-sera, it'll coat the CD5 positive cells and then macrophages can eat them. So you can destroy cells that way. So CD5 expressing cells, if you eliminate them, autoimmune disease. CD45 low cells, you eliminate them, also autoimmune disease. So what did Sakaguchi do with this information? ¿Por qué no los dos? Apply a serum that selects for both features at the same time. Problem is that you can't select for low amounts of protein, right? It's a good idea in theory, right? Why not both is correct. Why not both? And that is the key Nobel winning insight. But I want a little more, right? Sakaguchi combined these two pieces of information, but what do you think, or rather, what properties of his suppressor T cell do you think he was looking for? I think that's a good way to put it. What was Sakaguchi looking for in a cell that could be the suppressor? Could the suppressor T cells be using CD45 and CD5 as shortcuts to do their job? Don't think that concretely yet. Don't think that concretely yet. Stay back in the realm of abstract. Again, it's hard because I think it's actually easier to think about concrete things than it is to think about abstract things. And it's really more helpful to think abstractly first, so that you can reach these kinds of insights. T cells which had both CD5 and CD45? Not quite. Remember, it's CD5 high. If you eliminate them, that's autoimmune. And then CD45 low, if you eliminate them, then it's autoimmune, right? So it's not quite both CD5 and CD45, but you're getting closer. Yeah, so Sakaguchi combined two pieces of information. The first piece of information is when you eliminate T cells that have high amounts of CD5, you get autoimmunity. When you eliminate cells that have a low amount of CD45, you also get autoimmunity. So what did Sakaguchi think to do with those two pieces of information? What do you think? And I'm going to give you another bit of time to ponder it. Because again, this is why Sakaguchi wins this prize, is this insight. CD5 positive cells, eliminate them, you get autoimmunity. CD45 negative cells, eliminate them and you get autoimmunity. Man, this is hard. Yeah, it is. It is tricky, but I want to give you enough time because you can do it. You can do it. You can put it together. You can put it together. Take both and move them onto something with no thymus. Again, we're going to keep with the abstract. Don't think concretely. Don't think about things that you'll do. Just think about theoretical stuff. Theoretical stuff. What properties of the suppressor cell is Sakaguchi looking for? That's the way I'll ask that question. What properties is Sakaguchi looking for, given bullet point number 2 here? And I'll take one more sip and then I'll reveal the Nobel Prize winning insight, which, is simple enough where you're like, dang, if, I wish I came up with it because it is that simple. All right, here is the answer. Sakaguchi combined his, not really failure, but his 1985 paper with this other paper that found low CD45. And he asked this very crucial question. And the previous findings gave him a filter to sort cells by. What protein marker correlates with high CD5 and is inversely correlated with CD45, right? Sakaguchi might have been bummed out in 1985 that he didn't find the T-cell marker, but I bet he's really happy in 1995 because it gives him this amazing filter, because you can apply a stricter filter. So you can filter for proteins that are high with CD5 and low with CD45. And like I thought in the beginning of stream, a lot of you in chat had come up to this conclusion yourself, given the same information Sakaguchi had. Now, Sakaguchi had to do more than you did, right? Sakaguchi had to read everything and figure out what pieces to ignore and what pieces to deliberate on, whereas I gave you only the relevant information. I cleared all the noise out for you. But I just wanted to show you that you can have Nobel Prize level insight with a little bit of help as well. And that's what makes these so incredible, right? It is frustrating how elegant and brilliant this is, right? Because this is not a hard idea. Even though Sakaguchi is brilliant, brilliant has a form, and brilliant's form is simplicity. If you get caught up in the weeds and in the mechanisms and think too concretely, you often miss these brilliant insights. Because from here, then you can think, okay, if I filter by this criteria, how do I do that? That's a question you ask later when you think concretely. First, you think abstractly, and then you ask, how can I do that? And I think that is truly, truly wonderful. What do we want to prove with this suppressor cell? How is this hypothetical cell defined? Exactly. We want to find the identity. We know that this suppressor cell probably exists, but we don't know how to find it. Right? Like again, like I bring up the Higgs boson. Like in the math of the standard model, surely the Higgs boson must be correct because the math demands it to exist. But we, you know, until you measure it, you don't really have the proof that it actually exists. I actually don't know why the thymus atrophies with age. This is organ level biology, which I'm not too familiar with. There are protein markers, not the CD proteins themselves. There are protein markers that are not only CDs, but CDs are like a lot of them. Yeah. Why not both? Plus a different protein that correlates too. I mean, that is the approach that Sakaguchi takes, and isn't that cool? What do you think, chat? Do you get what I mean by like the form of brilliance is simplicity? Because it is this insight that enables Sakaguchi to actually design an experiment to find this T cell. A surprising amount of brilliance is rooted in not traditional intelligence, but in humility. Yeah, I mean, in chat, you all came to the same conclusion as Sakaguchi came to, right? If you have two pieces of information, you just combine it. I mean, yeah, Sakaguchi had to ignore a lot of stuff too, but these two were really, really important in the approach, the design of the approach. Yeah. So I think this is wonderful. I think this is wonderful, right? Because it is simple and you don't have to have a biology background to put this together. In fact, a biology background might harm you because you know more and so you can think more concretely, and that is occasionally a weakness, because if you can't also think abstractly, you might not be able to problem solve your way into solving this problem. Okay, so we have a filter. Let's try to find cells that have a lot of CD5 and not a lot of CD45, and then see what other proteins they express. And they have a technology for this. Marlon kind of brought up this microfluidics approach, and this is very similar. This is a technique called flow cytometry. It's a fancy technique, but it's actually not that fancy at all. It's a fancy sounding technique that you might find in like a sci-fi movie, but this is really, really simple. So you have a tube, and you have a tube that is very, very tiny. In fact, it is so tiny, it only allows one cell at a time. So let's say you take some blood from an animal and then you filter it through this machine. This machine will only take one cell at a time. It flows one cell at a time, and these cells have been coated with antibodies. These antibodies are against all of the CD proteins that they can attempt to find. Right, so CD45, CD5, CD4, CD3, etc., etc. You use antibodies against those proteins, and then those antibodies are conjugated or attached to fluorescent molecules. And so you get a color readout because as these cells are flowing through the chamber, a laser will excite the fluorescent fluorophores, and a detector will measure the specific light coming off of each cell. So every cell that passes through the flow cytometer will give the detector a unique fingerprint on the proteins that are expressed on the outside. Does that kind of make sense? I don't want to move on until chat is comfortable with that idea, the idea of flow cytometry. So I'll go ahead and start a poll. Just let me know if that idea makes sense. Go ahead and give me an idea. Right, so one cell flows at a time through this chamber, and the detector is getting a read using fluorescence to identify them, right? Every cell will have a unique combination of light that bounces off of them, and the detector will be able to identify what that type is. Once you find those new correlative markers, you can do more experiments on those new markers, really honing in on a unique marker. Exactly. What does CD stand for here? Oh, sorry. So CD is cluster of differentiation. Don't worry too much about it. Just think of them as proteins that are commonly used to identify immune cells. Sounds like running cells through a CT scan and comparing the values. Kind of. You can think of it that way. Like a single file line, and you're coded in a unique combination of fluorescence that the detector can detect. Okay, so does that make sense, chat? single file line unique combination of antibodies that are against these unique proteins on the T cell surface. Didn't hear the question? Yeah. So I'll go ahead and repeat it. Do you understand basically how the flow cytometer works? And I'll explain it one more time. You take a sample of cells, you incubate that sample of cells with antibodies against whatever you want. In this case, Sakaguchi is using antibodies against the CD proteins, which are immune cell surface markers. Those proteins, those antibodies are attached to fluorescent molecules, and then you run those cells single file down a tube where a laser can then excite some light, which then goes to a detector. And so every single cell that passes through the detector is going to have a unique light signature that tells you how much protein of what kinds are on the cell surface. All right, so it looks like we have a majority saying that they understand the flow cytometer. So I'll go ahead and continue here. So using this filter, the flow cytometer can allow you to ignore a criteria that you set. So Sakaguchi is filtering these cells one by one through this tube. The unique light signatures are coming through the detector. He can detect the cells that are high in CD5 and low in CD45. High in CD5 and low in CD45. So if you are not in this criteria, Sakaguchi can go ahead and ignore you. And if you are in this category, Sakaguchi can analyze you further. So what other CD proteins are you making on the surface? So if Caterpie is the only Pokemon that learns String Shot, you can filter by Pokemon that learn String Shot. I think that's, or the, okay, so Caterpie I think is not a good example. What's a good example? Gen 1, no, Psystrike is later. Really pulling on my Pokemon knowledge. Gosh. I can't think of any signature moves that are actually that unique anymore, because a lot of signature moves got the ability to be taught to anybody. I guess if a Pokemon knows Sketch, you can be sure that it's Smeargle, and if it knows the unique combination of, gosh. Yeah, Smeargle kind of just ruins everything because Smeargle can learn everything. So Pokemon is not maybe a perfect analogy, but it is similar, right? Like if you have a certain moveset that is unique, you can identify that Pokemon by its moveset. Okay. So Sakaguchi is filtering for correlates of a CD marker that he's fishing for. So what does he find? He finds that in T cells that have high CD5 and low CD45, they also have high CD25. So this protein is the protein that emerges as the candidate. Spatial rend is Palkia? Yeah. So if a Pokemon has spatial rend, you can be sure that it's Palkia. That's kind of the logic used. I think that's a great analogy, Marlon. So CD25 emerges as a candidate. So there is a T cell population that is high in CD3, which means it's a T cell, so that's good. High in CD4 means it's a helper T cell, so this is like a subset of helper T cells. And it's also high in CD5, which is correlated with successful suppression, and low in CD45, which is correlated with failed suppression. So you can see there's already quite a few filters on here. You need to be a T cell. You need to be a helper T cell, because previous research has shown that the suppressor T cell was likely a helper T cell variant. These are the two filters that Sakaguchi added on, right? High in CD5 and low in CD45. And the only protein that emerges here, the unique protein that emerges here, is CD25. And this is a lot cleaner, right? Because CD5, this is a protein that is associated with all sorts of different T cells. But using this filter, CD25 is the protein that pops up. And he's so lucky because that, there could be an alternate biology universe where there's not just one protein that is the T cell suppressor. Right, he got lucky. That's the other thing, is Sakaguchi had a brilliant insight, but he also got really lucky that suppressor T cells have a unique protein modifier. T plus helper plus suppression plus no not suppression. Yes, that's exactly the filter. And when you filter by that much, you get a pretty good candidate for the suppressor cell. So once you have a candidate marker for the suppressor's T cell, what do? So now, chat, now you can think concretely. Before I was ragging on you for thinking too concretely. Now we've done the theoretical work. Now we can do the experimental work. We've identified the T cell marker. So what do you do here? What do you do here? But how do they identify CD25? Like, how do they know it even exists? Antibody stuff. So this is foundational work that happened before, right? So they know that there are a lot of T cell markers, but they don't necessarily know what they do. So CD25 was just, oh, this is just something that T cells make. We don't know what it does, so we're just going to call it CD25 and raise an antibody against it and have all the tools we need to interact with it. It starts to sound like a bunch of Boolean logic. Exactly, it is. It is. Flow cytometry and cell identification is all Boolean logic, pretty much. Put it into mice sans thymus. If you can isolate it, definitely. Create antigens against it. Yeah. You can try to kill it, right? What happens if you try to kill CD25 cells? Bonk CD25 positive cells and see if autoimmunity blows up. Exactly. You can kill. Using the same approach that Sakaguchi has read about and performed himself, you can kill just the CD25 cells and see if that causes autoimmunity. And because the filter was more strict, you have a better candidate for the actual suppressor cell. I say knockouts too. We want to avoid knockouts at this time period just because CRISPR isn't yet a thing. Knockouts get a lot easier in mice when CRISPR happens, and it's still pretty hard at this time. It's not impossible. It's just you would have to spend a lot of money. So, Seed, you are right. That is something that I'm sure happened afterwards, but that's not what Sakaguchi did because in the past, that was a lot harder to do. It was one of the tool restrictions that you could not do a mammalian knockout that easily back then. That's why CRISPR was so revolutionary, right? Like we take for granted, oh, we just knock out things in the grand old year 2025. That's because CRISPR is so easy. CRISPR's so easy. What are knockouts? So knockouts are animals or microbes where you destroy a particular gene. So where Seed is getting at is you could destroy the gene for CD25 and see if that gives you autoimmunity. And I think that would be very interesting. And I think that would be very fruitful. It's just at the time, they didn't have the ability to do that. I love how so much of bio is just, what if we unplug this thing? Exactly. Like biology, I think it doesn't require that much hard skill, like hard math. Like that's why I think a lot of people who don't like math, but like STEM go into biology. Like the entry point is a lot sooner. Like, okay, you have to survive your college classes, but then you get to do new things very, very quickly, right? You can have new ideas in biology, I think, much earlier in your training than you can have new ideas in physics, at least from my perspective. Maybe physicists can prove me wrong, but I think to get a new idea in physics, dude, you have to learn all the math and everything that came before. Whereas biology, you need to learn the canonical biology and then just drill down into the problem that you are looking for. And maybe physicists would say the same thing about biology. I don't really know. Chemistry, yeah. Okay, so let's try to break the CD25 T cells using the same approach, right? So Sakaguchi raised an anti-CD25 sera against CD25 to specifically kill these particular T cells. So if CD25 positive T cells are in fact the suppressor, then these mice should develop autoimmune disease. And in fact, they do. I wish I could show you the data, but they do. So they also, here's another benefit of doing this, they develop a pretty severe systemic autoimmune disease as well. So it's not these like, targeted, they have one organ that is under attack, like these weird edge cases that they were seeing before. If you knock out CD25, the mice develop like incredible autoimmune disorder, like very, very quickly deadly. The body turns on itself, eats itself very, very quickly if you do not have these regulatory T cells. And you can imagine, right? Like filtering a lot of blood day in and day out, you need this regulatory T cell to keep the peace. Otherwise, just the volume of protein that your T cells come into contact with, they will start to learn incorrectly, which just shows you how important this cell is. The fact that if you kill this T cell, the mice degenerate very, very fast, right? This is an extremely important cell to make sure you just survive. So Sakaguchi showed that the CD25 positive cells are necessary to protect against autoimmunity, right? When you remove them by means of antisera, you get autoimmunity. So what's the other half of this equation? And if you are a biologist, hold your answer. But if you are a stream watcher and not a biologist, this is a test to see how many of my streams you have watched, right? So Sakaguchi showed that CD25 cells are necessary to protect against autoimmunity. What's the other half here? They are necessary, but are they? And it starts with an S. See if anybody can get it. While I hydrate real quick. Right, they are necessary, but are they blank? Are they blank? And this is a very, very important paradigm in biology. A lot of foundational molecular and cell biology is built on this very simple idea that you need to test for necessity. And ideally, you should also, if you can, test for something else. Do the MCAT cells in the thymus not function without the T? Oh, the cells that teach the T cells. I don't actually know how they interact. That's a little deep for me. Yeah, all right, we've got the answer in chat. So this paradigm in biology, extremely important. I think important for any like hard science field is testing for necessity and also sufficiency, right? Are they sufficient to protect against autoimmunity? So if you remove the CD25 cell, does it cause autoimmune disorders? But if you put back the CD25 cells, does that protect against autoimmunity? If you can prove both of these things, you have an excellent candidate for the actual autoimmune protective cell, right? You need both sides of this coin to show how important this cell actually is for this biology. Okay. So how do you actually do this, right? How do you give a mouse, or how do you rig this experiment up? So what Sakaguchi did was he inoculated these nude mice. These mice are mutated, where they do not have a functioning immune system. I don't know how these mice were generated, but just know that they do not have a functioning immune system. They have to be raised in germ-free environments. So if you give these mice T cells that do not have CD25, you filter by CD25, they have exacerbated autoimmunity, right? So they lack a functioning thymus, and they are functionally without an adaptive immune response, right? They don't have like functioning T and B cells. But if you give them every T cell except the suppressor cells, right? You eliminate only the CD25 cells, it caused autoimmune disorders. And that's interesting, isn't it? Why do you think they couldn't give them CD25 positive T cells? It makes sense. It makes sense, doesn't it? Right? A lot of biology is very accessible if you keep all the acronyms straight. Right? You give them everything but the CD25 cells, and then it causes an autoimmune response. Yeah. So they narrowed down that there's something in the T cell population that is suppressing the autoimmune response. And when you give everything but the CD25, you successfully cause an autoimmune reaction. It's a lot of biology is Boolean logic, right? If then, if not, or nor. Get real comfortable with thinking logically that way. Yeah. Go ahead and take another drink. So this is an indirect way to say that CD25 is sufficient, right? Because they didn't actually do that experiment, I think, right? They didn't inject with CD25 positive cells because I think they're just experimentally limited, right? Oh, actually, maybe they did. I don't remember how they did this experiment, but I think in that paper they also managed to purify CD25. But I think Oh, wait. Co-inoculation prevented autoimmunity. Right. Because I don't think they actually have a means to purify CD25 only. I mean, chat, you're right in that the perfect experiment would be give a mouse with an autoimmune disease CD25 cells only and then see if they get better. But I think that purifying this particular cell at this stage in biology was pretty hard. So what they did was they inoculated all the cells besides the suppressor, which they know causes an autoimmune reaction. And then at the same time, they put back in the CD25, and then that stopped the autoimmunity. Right, so they're not able to purify just the CD25, but if you give them both, you prevent the autoimmunity. That's incredible, isn't it? Right, another one of these logic gate things. And I want to sit on that for a bit so that you can kind of digest that idea. That, like, chat's idea of give an autoimmune reacting mice CD25, that would definitely work if Sakaguchi could do it. But instead, he had to take this little more roundabout way where first you prove that everything minus CD25 causes autoimmune. And then when you add back in CD25, then you remove autoimmune, right? Does that make sense? Does that make sense? It might take a while. If you get deep into biology, you get very used to thinking this way. But if you're not used to thinking about breaking things in this way, it's kind of hard. Oh, good. Yeah, it's wonderfully intuitive, right? Wonderfully intuitive. It's a little not as straightforward, but that's because technology limitations. And this is a useful tool that you can take with you in your scientific journey, because this is how Sakaguchi worked around the fact that I don't think that they could have purified CD25 only. And yeah, the CD25 inoculation wouldn't work if it wasn't specifically induced first. That's also true, Marlon. Like, you first have to give the mice an autoimmune disease first. But this, like, structured plus minus proof, it's like very mathematical, very satisfying. Both the CD25 minus and CD25 plus has to come from the same source to make sense, right? So it doesn't make sense to inject CD25 plus into something suffering from its own CD25. I mean, they do it to check whether or not they can stop an autoimmune reaction from happening, right? They're looking for the cessation of autoimmunity, not the induction of autoimmunity. Yeah. I watch your vids to get better at experimental design. Thank you. That's a huge compliment. I mean, I was, before I did YouTube full time, I did teach a lab course, so I'm glad that I can keep some of that, that ethos alive. Question: where do the adaptive immune cells go after they differentiate then, if not the thymus? Some of them live in residential tissue. Like, you have residential T cells as well. I'm not an expert in that too, so I'm not going to be able to say much more than the fact that there are resident B and T cells that live peripherally. Weird question. Could one become autoimmune against CD25? I mean, in biology, a lot of things are possible. Likely is a different question. Hypothetically, the answer is yes. But whether or not it is practical, who knows? Oh, they go into lymph nodes. Ah, that's absolutely correct, right? That's why they're called lymphocytes. I just had a brain fart. Yeah, so that's why we check for cessation before we get into the logic of whether it is possible to cure. Yeah, exactly. Okay, so incredible, smart design, Nobel Prize winning, worthy. Okay, so Sakaguchi shows that CD25 positive T cells are both necessary and sufficient to promote autoimmunity. This is a very, very good case that this is the T cell suppressor. So T cells are necessary because when you destroy them, autoimmunity happens. They're sufficient because when you add them to a system with autoimmunity already happening, autoimmunity decreases, right? And both of these properties, combined with the fact that this is a unique marker that allows you to filter everything else out. Now, does this mean that no other cell makes CD25? No. What this means is that the unique combination of CD3, CD4, CD25, CD5, and low CD45 is the filter that gives you the identity, the sure identity of the suppressor cell. While other cell types may play a role in regulating autoimmunity, Sakaguchi lands on the first major convincing identifier for the suppressor T cell. Sufficient doesn't always mean the only thing involved ever. It just means that it does not need to be enabled by something else. So sufficiency is a very genetics level term where, you could have a gene that causes a disease, but it only causes the disease when you destroy a different gene as well. Like it's synergistic. What sufficiency tells you is like how important this thing is to the phenomenon you're studying in a vague, indirect way. So independent, exactly, Ari. Yeah, it is independent. It doesn't need another mutation or another thing to show off its worth. It is sufficient and necessary. So both of those facts, sufficiency and necessity, and the fact that the combination of unique cell identifiers, all of these provide us with the world's first real identification. You can see that Sakaguchi recovers from failing 10 years before, right? In 1985, Sakaguchi thought maybe it's CD5 and saw that CD5 was okay. He's narrowed it down a little bit, but not enough. And then combining it with the other group's work showing that CD45 low was a criteria, he took another shot at this problem and found the cell, the suppressor cell. There are other neat things about this paper, but that's all the time I have for now. To give you a sneak peek, this paper also looked at skin graft rejection and foreign antigen rejection, which showed that the CD25 cell is not only important for autoimmunity, but heightens it also contributes to dampening the response against skin grafts and bovine serum albumin, which is just a random cow protein, right? That you inject to a mouse to see if it can mount an effective immune response against a foreign antigen. And what they found was that these CD25 cells were not only autoimmune cells, or rather autoimmune regulating cells, they were capable of dampening the immune response as a whole, right? So it's not just autoimmunity, it is regulating the immune system entirely. Okay. It's a neat paper overall, and actually for a paper that's that old, surprisingly readable, right? The further back you go in history, the worse these papers get. And Sakaguchi also, for being a Japanese scientist, the English is quite well written. If you can get access to this paper, I would recommend that you read it. If you're a biology student, if you're not a biology student, you can certainly give it a try. But if you are a biology student, this paper is pretty readable, I think, if you're an undergraduate. So I recommend that you give it a shot. What's the paper? I should have had it linked, but I don't. So instead, I'm just going to tab back to what the paper is called. Sorry, I'm going to go all the way back at the beginning. It's got a long title, though. That's what I'm kind of annoyed by, but... Oh my god, it really is like... Okay, here we go. Immunologic self-tolerance maintained by activated T cells expressing IL-2 receptor alpha chains, aka CD25. Oh, the description is wrong. Oh, I didn't even realize I had a thing in the description. Oh, it still has the mitochondrial sleep study. Ah, my bad. Okay. I'm going to real quick tab all the way back to the end here. And so I can wrap up this stream. Whoa, went too far. Let's see, can I easily get this back on screen? I can. Okay, yeah. So what's next for the T regulatory cell? The mechanism is still very, very not well understood. Maybe you'll be the one to discover it, right? This is for your generation. Despite it being known for the last 20 years, 20 years? 30 years, oh my goodness, 30 years. We don't really know a lot about how the T regulatory system works. We do know that it is important in the graft versus host disease, so if you're interested in studying organ transplantation, this is going to make an appearance in your studies, in autoimmune disorders, in cancer, in infection as well. There are all sorts of cancers and infections that modify the T regulatory cell to make them stronger so that they can suppress the immune system. Super, super interesting interactions. And for my channel, I talked about the T regulatory cells a little bit in my gut microbiome video. So the Treg is like everywhere. So you can imagine this is a very, very crucial discovery in the realm of immunology and medicine. Very, very cool. Most colleges give you a way to legally access them. Otherwise, there are other ways to access them for those of you who aren't a little afraid of. That is fair. That is fair. Okay. So that is all I have for this Nobel Prize stream. Keep your eye out. I believe on Sunday I'm going to release this plague video. I'm just waiting on getting my merch, right? One of my charity stream incentives at $1,000, which thank you all very much, by the way, for helping me raise $1,000 for glioblastoma for St. Jude's Hospital. But because you have enabled me to reach that goal, I will fulfill my promise delivering merch to all of you. It's just that my merch shipment got delayed and I want to double check the quality of the merch before actually putting it out there. So unfortunately that has delayed the release of this video, which I worked really hard on. But keep your eye out on Sunday for the release of this video, the plague video, Yersinia pestis. Very, very exciting. And with that, I will take questions, comments about biology, about the Nobel Prize discovery, and I'll try to answer them as best I can. You can let me know how your school year is going if you're a student, or, I like to leave this last bit of stream to free chat before I go off and have my dinner. All right, thank you all for attending, especially those of you who have stayed up through your incompatible time zones to watch me yap. I do try to tackle a couple of different time zones because I don't think my science content should be gated to people who live in the most friendly time zone for me. As long as you can tolerate the sound of leaf cutters or whatever's going on outside occasionally in this time slot, because it is morning where I am. It is morning where I am. Black Death video? Exactly. It is the Black Death video. It is the plague video. Thank you for all your hard work in preparing this presentation shows. Thank you so much for attending. I do it because you all show up, because I believe I am providing value. If I didn't think that I was providing value, I wouldn't be doing them, which also means that you should let me know if I am providing value. I'm so excited to see your video. They're also well made. I absolutely love the road roller from the white blood cell video. Yeah, I loved making the neutrophil video. It's the best video I've ever made, but YouTube's algorithm just didn't like it for some reason, which I'm very sad about, right? I think that's probably the best video I will make in a very long time. It just has so, like, the gags per minute in that video are so high. Like, I felt so... The thing is that when you make a video about diseases, you can't really be that funny. Because you don't want to joke about people dying, right? And a lot of the diseases I cover are pretty horrible. But with the white blood cell video, I was free to be as funny and weird as I wanted to be. Still suffering through the unemployment grind set. Yeah, me too. Unless you count YouTube, but it's not quite there yet. Hopefully with merch out, it makes YouTube a bit more of a viable career option. But yeah, much sympathy to all of the bros who are undergoing the unemployment grind set. That sleep study was a pain. Yeah. The mitochondrial sleep. Yeah. That's tough. That's a bit more abstract. What I like about doing these Nobel Prize streams is, like, the papers written in the past were a lot harder to read because the English is often not very good. Although, again, Sakaguchi's paper is really well written. You should definitely read it. But the experiments were a lot simpler. These days, the technology is so advanced that it can get kind of tricky to walk through everything here. It's going okay. Ecology is so-so. Biochem is neat. Struggling with chores. I hate sweeping. That is fair. That is fair. Since becoming a full-time YouTuber, chores have become part of my actual work, right? It's become like a mini game to try to optimize the time and effort it takes to do them. And if you view it that way, maybe it gets less annoying. Like, how do I optimize my how do I chores max? I enjoyed the stream. It's a shame that I missed so much of the game. Oh, it's totally okay. I was really looking forward to watching your takes on it. Well, there's always the VOD. Yeah, and life does happen, and I'm sorry that you had to address that. You give me so much motivation for studies. I got no one else to geek with. That's good. I'm here to provide that space for you to express the love for learning sincerely, right? Because I think there aren't that many places online where you can do that, in a live format, right? Of course there are wonderful YouTubers out there who do content, but I think there's like an energy when you're all trying to learn something together, right? It makes it less lonely, I would say. Especially when I'm not calling you out by name, right? Because in graduate school, that made learning very unfun. To be cold called was very, very scary. But when you are all anonymous and participation is opt in, I think it garners more affection, I think. Algorithm be damned if I ever get a social handle. Since my theme allows me to, I'll have no specific field I'm limited to. I'm going to do what I can to get the word out there. Yeah. I mean, traditional methods of marketing yourself, not relying on the algorithm, are still very valid. Just, I feel like our virtual YouTuber subreddit doesn't really want to upvote, who I am, right? I find that it is often very discouraging to put myself out there when I think, even though I count myself as a virtual YouTuber, I don't really feel like I belong in that kind of way. And that's a little sad, but I am happy that the algorithm has approved of some of my videos. I'm just going to have to find my audience through there as opposed to places like Reddit or Twitter. Or maybe it's me, right? Maybe I give off a vibe of like coldness that people don't like. Because, speaking behind the scenes like V-Theorem, there'll occasionally be people like, oh, hey, so-and-so like reached out to me. And I'm like, no one reaches out to me. No one wants to be my Twitter friend or anything, which I think is sad. I think part of why, what inspired V-Theorem is this idea that like I felt like an outsider, and if I feel like an outsider, well, I guess I'll just have to make my own friends, right? And that's where V-Theorem came from. Reddit isn't the nicest of places. I wouldn't say that Reddit isn't nice. I just think that, maybe I'm not good at marketing myself on Reddit. Maybe I don't know the combination of words to say, or maybe the people who are on virtual YouTubers are not looking to engage with who I am, right? That's also possible. And it is something that I have learned to just have to live with, because I don't want to be a traditional looking VTuber, because I don't actually identify with manifesting as an anime boy. Right? I don't actually want to be an anime boy. There actually is a VTuber model that Toxo has yet to make public of me as an anime boy. And, back and forth, we just thought that I think my personality fits more with Phi as a neutrophil than as a traditional VTuber. That's a valid point. I wonder what makes people feel concerned about reaching out to you. It's not that no people have reached out to me. I think there have been a brave few, but I think like given the disparity, like I mean, I'm I don't want to. There are some people in V-Theorem who get reached out to a lot. I'm like, nobody wants to talk to me. But I Frost, I think you might be right. Like, I think the way that I carry myself might be a little more intimidating in the way that I present more authoritative as opposed to friendly. But I don't know. I think I come across as, I hope I come across as pretty friendly. Like, I just, maybe it's because most of my content is educational that people don't feel comfortable reaching out. But I actually, like, if you make content, I guess here's the thing. If you make content and you're interested in talking about content, I'm generally pretty open, and you can take advantage of it now while I'm still not Mr. Beast level, right? Like once again, I'm full of myself, so I think once I become successful, I don't think if I become successful, I think when I am able to go full time, right? That's just the mindset I have, and I kind of have to have that level of delusion to want to do YouTube full time, because if you don't delude yourself into thinking you can do this full time, you'll just give up without ever giving it a try. So even though I know it's extremely unrealistic, there is some preliminary data that it is possible, and so I must believe that it is possible, and I must not stop trying until it breaks me, right? But once I do find that success, it might be harder to talk. So if you make content, we can have a chat about content or education and stuff like that. My only line is that if you don't make content, that's where I like draw the line in terms of like answering messages, right? I don't do that. Except if you are a fellow content creator. But it doesn't mean I won't talk to you. It's just not in private, right? It's like I have a Discord. We can chat in the Discord. Hopefully that makes sense. You are the hottest anime boy. I think so. I think anime boy V2. This is my toxic trait. Is that I think I don't identify, because most anime VTuber models that get attention are like super conventionally attractive, and I don't want to be a conventionally attractive anime boy on the internet. I don't feel that way. That's not a reflection of my inner self. It doesn't align. It'd be like identifying with something that isn't me. You know, you can enjoy all the hot anime boys you want, but I don't want to be looked at like a hot anime boy, and therefore I do not manifest as a hot anime boy. Yeah. I'm curious as to why mice with no thymus attack their ovaries first. Is the immune system just more sensitive to gametes? Here's my complete guess. Okay, complete guess. I don't know the answer. Okay, let me be very clear. I don't know the answer. My guess is that the reproductive system in females is immunologically funky. And I think this is because when you are growing a baby inside of you, the baby is not you. And so there have to be certain exceptions to how strong the immune response can happen, right? Like you don't want a strong immune response near the reproductive tract because, you know, the whole thing about selection is that it's pretty good to pass on your genes to the next generation. And so my guess is that the area in the ovary is somehow immunologically distinct, that it is unique in some kind of way because of its unique role in human biology, or in mammalian biology, I should say. I think it's also that you're not on Twitch, which I think it makes it harder to organically get onto your streams. Yeah, but Twitch, I just don't like, man. Twitch is so hyperactive, and I think my vibe is a lot more chill. Like redeems and stuff like that. I would have like no redeems. Like I don't want to do redemptions on Twitch, and that's part of the whole culture on Twitch, which I don't like. But I do like raiding on Twitch. Raiding on Twitch is so easy and comfortable, and it makes it so easy to make connections. On YouTube it is a lot harder. That is true. Because on YouTube you can only raid people that you're subscribed to. I mean, you can raid other people, but the only way to make it easy is to raid into somebody that you already know, which, by the way, maybe I should try to make an attempt. The thing is that there aren't that many. Because could I raid into like big YouTubers? Sure, I could raid into big YouTubers, but I don't want to, because it's not productive, right? They're not gonna care who I am. We're not gonna like establish a connection in any kind of way. So why would I raid into someone much bigger than me? Whereas, you know, someone who is more comparable in size, then we can actually like establish a connection and a community. Which reminds me, I should set up the raid for this stream. Is there somebody streaming right now that I can raid into? The answer is so frequently no. And the other thing is, I would prefer to raid into people doing science content instead of people just doing gaming. And it's like very rare that people like do live science content, which is like sad. So instead, let me just set that up there. I think we will raid into Penbu's 2-year anniversary, which isn't happening until tomorrow, but the redirect will give you the link at the very least. Yeah. So if you know any like VTubers who do science-y content, then you should let me know, because I would love to raid into them. The subscription feed is always so lonely, I think. Science history and literature teachers should work together, because I think it could help students to learn better. I think having a well-rounded education is very, very important. Like the hubris that comes from STEM, thinking that they don't need to study the humanities, is responsible for, I think, a number of adaptations of technology that have made life worse in a lot of ways. We'll put it that way. But respect is that no matter what, you want to make the kind of content you want to make. I'm incapable of selling out, in the sense that I've tried to make a couple of you know, sell out videos. Not like, I mean, that's a harsh term for it, right? Like I tried to make videos that I thought would appeal to the algorithm and they bombed. Not only because I think I just didn't have an understanding of what the audience wanted, but also like, I don't know, I just didn't love them the way that I love some of my other videos, right? I think the common example is like the kombucha video I did, which I was like, I'm gonna put Brad Leone in the thumbnail, you know, because he made kombucha one time and people will click on Brad's face, and then like nobody clicked on it. It was not a great showing for a video, even though I think the video itself was okay. But since then, I'm just like, I'm only gonna make content that I'm actively passionate about, and just hope that there are enough of you out there that the algorithm can loop in to find me. If people value you for your attractiveness, that means they're not valuing as much for what you bring to the table or something to that effect. Yeah, like I don't want the attraction as like an influence, right? I think the experience that I would, and you know, if I wanted success, then of course I would do that, right? Of course, having an attractive model is going to make number bigger. But I didn't want that to, but I didn't want to attract an audience who was watching me for that kind of aesthetic, right? I would much rather have an audience who sticks with me through thick and thin because of what I do and not because of how I look, which is kind of interesting because as a VTuber, you can choose to look like anything, right? Like if you want to be a handsome anime boy, you can do that with this technology, and I wouldn't blame you for it. It's just not, it's not, I don't want to feel like I have that expectation to live up to, I should say. So is this why STIs are so dangerous? That's a good question. I don't know. That's interesting. My intuition would say that it is more because it is a warm place outside of the body, kind of outside of the body, that you can't mount a crazy immune response to. Actually, that is what you're saying, isn't it? Yeah, I think that's probably pretty reasonable, that the immune response is a bit different within the genitalia. A lot of major VTubers from agencies have no redeems and just stream across multiple platforms and do their own thing. I thought about multi-streaming on Twitch, but I feel bad because my members who pay me money are on YouTube, right? I don't want to make their experience of my content worse by having to divert attention to Twitch as well. Even though I think Twitch's like culture to support streamers is different, I do think that I would, for now, like honor the members that I have on YouTube, because I don't want to be making content that is like, if you subscribe to me on Twitch, just don't. First, Twitch takes more money than YouTube does, and I'm not going to offer Twitch viewers any more than I would offer YouTube viewers. In fact, they get less because YouTube membership gives you access to my non-science content. So there are a lot of reasons why I don't want to go to Twitch, but it is very, 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 very tempting. I know some science VTubers who use YouTube for education and Twitch for gameplays. Yeah, I know, what's her name? Shioripon, the surgeon VTuber, lovely streamer, uses Twitch for gameplay and then YouTube for her medical education streams, which are awesome. I'm in there all the time. Thing is I don't do gameplay content. I tried to like do the gaming thing. I just don't have as much fun as I do yapping to all of you about science. I know a few from Brazil. Other than that, I only know V-Theorem and friends. Yeah, I bet you know. I don't know how to speak Portuguese, but Biolojo Andy. And there's also who's the other guy? Dr. Echolis. Is that who you're referring to, Seed? I think they're Brazilian. I think. Speaking as an engineer, please support your local philosophy department. They do important work and they're very important to teaching ethics and critical thinking, and their work is foundational. Yes, exactly. Like STEM can help you build the thing. Humanities tells you how to best implement it for the good of humanity. I'm working slowly towards my V1 debut. That's exciting once my real life is more stable. Also very relatable. I didn't start this until I had a stable life either. I'm the same in terms of wanting to be a non-human as a VTuber. Yeah, I think there's something very appealing about it. Something very, very appealing about it. Yeah, actually, let me promote Dr. Echolis for a bit, because he's an interesting guy. Let me see if I can pull up his info. Dr. Echolis, YouTube. So, oh man, why is your channel so hard to find? Ugh. Is this it? Man, I cannot find his channel. That is so sad. Maybe I'll put it in the stream description instead. Okay, see, okay, great, great. Yeah, it is Andy and Dr. Echolis. Put it in the Discord too. I will. But I can't find his freaking channel. I would like to link it right now, but oh, I was just spelling it wrong. Okay. So let's link Twitch here. See if I can do it, because I think YouTube automatically has like links blocked or something. But I'll put this in the Discord as well. So I don't know a lot about Andy. He's a bigger content creator, but Echolis, he for the last couple of years he's been doing like a VTuber science symposium where he's organized, and major props to him, and it's very hard to organize VTubers to do literally anything, and he's organized a couple of VTuber symposiums these last couple of years, which have been interesting to observe. And I definitely applaud the effort. Like, it's very impressive to be able to do that. Where I'm not saying that like there aren't other science VTubers out there. I'm just saying it's really hard to find them on YouTube specifically. So if you have recommendations, please let me know. Okay. Yeah, so I'm gonna plug Echolis only just because I haven't watched Andy's or Telhado's content all that much, so I can't like personally endorse them. But Dr. Echolis, I think I can say, has done some pretty cool stuff. And he was on the 2024 symposium. That sounds reasonable, because I think the first symposium he was there too. And that's very fun. And I love efforts like that. That's always so cool to me. All right, with that all being said, it is time for me to go. Thank you all for attending stream. I hope you learned that you too have the capability of being a Nobel Prize winner, provided you have the support, the effort, the grit, the insight, the foresight, the background, etc., etc. But yeah, again, their brains don't do anything different than yours. They've just been steeping it for longer and have accumulated more skills. And I hope that I don't know. To me, that makes me feel better, because when I was a kid, I was like, "Oh, these Nobel Prize, they're all geniuses. They're like a different species. They're not a different species. They're still human, but they are very hardworking, very skilled, very talented, and that is what I want to leave you with." Any plugs I can think of? My video comes out on Sunday. Merch also comes out on Sunday, provided that the merch is of decent quality, which I'm very excited about. And also, Penbu's second anniversary is coming up tomorrow. I will try to make it there to hang out for a little bit. Penbu, I've gotten the permission to tease this collaboration, but Penbu is going to be teaching me how to compose music at some point in the future. And if you've seen a little Easter egg on YouTube, there's a collab video that I'm involved with where I try my hand at songwriting. It's not uploaded on the channel or promoted anywhere. I'm just kind of seeing whether or not the YouTube algorithm can find it for you. But I've recently dabbled in making music for these videos, and Penbu is going to help me kind of figure that out. So if you want to see what he's about, he's going to be doing his anniversary tomorrow, and this stream will redirect to him. Okay, that's it. Bye, y'all. Bye-bye. Bye-bye.